This marks the 10th video in my challenge of 30 videos in 30 days, one video a day. In today's video, we're going to be examining what could have been the simplest life form produced by emergent chemical evolution, and basically by any origin of theory could hope to produce. So, my name's Eduardo Hernandez. I am the creator of Emergent Chemical Evolution, the newest theory on the origin of life, the most complete. I am also the author of the book by the same name. You can find it on all online sellers. So let's get on with the video. One of the simplest life forms that I like to go back to in describing in my book and in some of the molecular examples in my book about basic metabolic processes, simpler metabolic processes compared to human processes, is the E. coli. The E. coli, again, is a single cell organism that has one cell wall. It does not have mitochondria or a nucleus. It only has one chromosome, and it creates its biomolecules by forming smaller polypeptides to unite to make one larger enzyme to then do the metabolic processes. But that is not the simplest life form there exists. There are other life forms, other bacteria that are disease-causing bacteria that are almost as small as viruses. It's even simpler than the E. coli. Man, for the life of me, I couldn't remember. But it's very, very small. It has fragments of ribosomes. It has fragments of DNA in it. And it's very, very simple. But that being said, that is still a very complex system that would not have been produced immediately at the origin of life. Again, that organism, as small as it is and as simple as it is, has hundreds of genes in it. It has hundreds of genes to produce hundreds of different types of proteins. As simple as that bacteria is, in post-production and after editing, I'll put the name of the bacteria at the bottom of the screen. That bacteria is still way more complex than any simple origin of life environmental conditions could produce. That is just a level of complexity that is way above what any tenable system could have produced at the origin of life, at the primordial soup. Let's put it, because that's basically emergent chemical evolution is a primordial soup theory, but it better encapsulates the conditions that would have existed at that time, creating a conducive environment for the emergent chemical evolution to take place, but for the chaos engine to coalesce and start functioning. So again, as I said in previous video, the product of that engine would have been more complex polyamino peptides that would have been taken through cycles, but they would have been molecules that would work in concert to create biological reactions to occur. Now, if we're working backwards if the bacteria, the simplest bacteria that we know, has hundreds of genes, that would not have been produced at the origin of life. I find it untenable for any system to spontaneously emerge in an encapsulated bubble of fatty acids, creating a quote-unquote membrane. I find it, again, almost on the border of spontaneous, what is that thing called? What's that theory? Spontaneous generation of life forms, because in order for an encapsulated bubble to reverse engineer into it genes, hundreds of genes, in order for that system to continue to propagate, is untenable. There had to be a simpler way, which is what emergent chemical evolution describes. It is that components of a system would have been generated. Well, let's work backwards. What is the organism with the least amount of genes in it? Now, any bacteria, any cell, all of them, like I said, the most simplest that we know of, have hundreds of genes. So that disqualifies any bacteria or single unicellular organism. Viruses. Now, with the discovery of the Mimicked virus, which are megaviruses that have thousands of genes and very complex mechanisms working within its protein code. Well, it's a mix. The membrane is a mixture of protein and fatty amino acids, but it is a simple membrane. It's not an active membrane like a unicellular organism. These viruses have genes that make enzymes that are unlike any enzymes found in any cellular organism. These viruses create metabolic proteins that are unlike any in any of the other kingdoms of life. So these viruses, to me, represent the pre-cellular stage of life. According to my theory, and here I'm going to say it, before cells came viruses. There's no way around it. Viruses need to exist inside other cells or conducive environment for them to propagate. Of course, we see that they hijack the components of cells to replicate themselves. But what we ignore is that they self-assemble. The conducive environment of inside a cell, like I've mentioned in other videos, inside a cell, the cytoplasm, is, if you look at the small section of a cytoplasm and you look at the conditions at the origins of life in a small section of the sea, the components, the pH gradients, which modern cells create pH gradients in order to help reproduce and maintain their homeostasis. Those conditions are what viruses need to propagate and to exist. So, but the important part is that these components of viruses, when you put them in the proper condition, they self-assemble without the need or the aid of other molecules. This is a very important behavior to have that would have very much benefited life at the beginning of life, at the origin of life. This behavior of viruses is not produced or mimicked by cellular organisms. They cannot. Once you destroy the membrane of a cellular organism, it cannot reassemble. You cannot just take the components of a cell, put them in a petri dish, and expect them to form a cell again. It doesn't work. But take the components, regardless of their source, albeit of the same virus, of course. But even if you get components from different cells, put them in a petri dish, they will, well, a petri dish with the proper environmental conditions, these molecules will find each other and self-assemble a virus. So to me, that screams of viruses being the gap between molecules and cellular life. They perfectly fit, being that they're so small and different levels of complexity, perfectly fit between molecules and cellular life. You have the Mimicked virus, which is a couple of steps from cellular life, being so large and being so genetically diverse with the amount of genes that they have. And then you go back in the other direction to simpler viruses. Now, one of the classic viruses that have been used to study genetic diversity, genetic mechanisms that was used was the TMV, the tobacco mosaic virus. This virus only has 4 genes. It has a gene to produce proteins, 2 proteins for its coat, and another protein, 2 other proteins to replicate its genetic information. And that's it. The important thing about this virus that was instrumental for me in solving the problem that would have occurred at the origin of life is this: the assembly and disassembly of this virus depends solely on the pH of the liquid, of the medium it finds itself. The pH has to be at a certain pH for the proteins to become rigid enough to then interact with each other and form lock and key structure, which is a long rod, and they form and encapsulate as they go. They encapsulate the RNA information necessary to reproduce the tobacco mosaic virus. It is the simplest virus, at least that I know of right now. It only has 4 genes. That is the simplest, to me, the simplest life form that exists. And it fits perfectly. In order for it to replicate, then, it must find itself in another pH level that then takes the protein coat out of its conformation, meaning it becomes not as rigid and doesn't hold each other as well, and they come apart, releasing the RNA, and that RNA then, finding itself in the proper cell, would then hijack the replicating systems of that cell and make more copies of itself, therefore creating hundreds, thousands, millions of copies of itself and exploding that cell. I mean, the fact that these organisms, viruses, can readily hijack the replicating systems of cells says something. These guys are replicating engines. They're basically unstoppable. That's a characteristics that would benefit life at the origin of life. So again, emergent chemical evolution through the chaos engine would have created multiple versions of enzymes and biological components that would have benefited any organism at that time and create an environment like that of a cell where viruses propagate. So this, to me, shows that viruses was the stopgap between molecules to cells. They are the perfect genetic genetic replication agent. I mean, no, the perfect genetic novelty creating agent. They reproduce so quickly. And one of the reasons why viruses are so contagious, so hard for our immunity to fight against, is because of the introduction of errors in the replication of viruses. When they reproduce so fast that there's so many errors, they evolve so quickly that our immunity cannot keep up. That's why we have the common cold. The common cold is a simple virus, and our body's able to fight them off most of the time. But it mutates so quickly that the next time we come in contact with it, it has changed so much that our immune system doesn't recognize it. That, to me, is a perfect novelty agent for the origin of life. These things would have reproduce so quickly and create so many errors that evolution would be boosted, would occur at such an accelerated rate that it would easily gain the genetic information to produce novel proteins that create beneficial molecules for life. Viruses would have easily created new novel systems for the encapsulation of RNA, the mechanisms for reproduction, the mechanisms for creating structures, more complex protein codes. And according to an article I read from one of the leading world virologists, he suggests that DNA was the product of viruses, that viruses created DNA from RNA through its mutations, through its novel, through this accelerated novel evolution, that DNA would have been invented by viruses and then introduced into cells, because he's working with the dogma, the latest theory of viruses originated our genetic junk from cells, which I absolutely disagree. Again, the TMV, the tobacco mosaic virus, is a perfect I gotta edit this out. It's a perfect model organism for showing the direction of evolution from molecules to cell. Let me explain. The tobacco mosaic virus seems very hard to have been able to de-evolve from fully functional viruses into this type of virus where it only has 4 genes, it only has the protein encapsulation to hold RNA. To me, the evolution would have been from a free-floating RNA into a protein-coated virus. Being that 99.99% of all organisms that ever existed have become extinct, should inform us of that a cellular organism losing capabilities and then de-evolving into viruses, or viruses being produced by replication error of cellular organisms, to me is untenable. To have the perfectly matching protein code to be able to encapsulate a certain amount of genetic information, insufficient genetic information, to then go off into the environment and exist on its own, is, to me, is untenable. What we see in cells is not possible. Cells are such a complex, synchronized machine that any error in its replication would lead to its eradication, not the production of simpler viruses. That, to me, is not how we've seen evolution work. Evolution works, it can create non-detrimental reduction in an organism's functions, but most of the time it leads to its extinction or its survival. You don't have anything in between. There's no evidence of that anywhere. But what is more tenable to me and more logically plausible is that molecules evolved into a more complex, albeit simpler, organism than cells. Let's get back to what this video is supposed to be about. So again, what we should expect from origin of life, from any system that would create the origin of life, is what everyone is looking for: RNA. I agree. There cannot be no disagreement. RNA is at the heart of life. Without RNA, you cannot produce proteins. Without proteins, you can have RNA. So how did that happen? How did that system come into place? The chaos engine in emergent chemical evolution perfectly explains that. What would we expect to emerge from any system, albeit, let's say, emergent chemical evolution is not real. Let's say one of the other theories is the actual thing that created the origin of life. What would we expect that system to produce? What they want, what all of us want, because it's essential to life, is the production of RNA. How that came about, let's ignore that portion for this video. Let's say theirs was correct. What would we expect to be produced? An RNA molecule with random nucleotides. An RNA molecule that does not have any information to produce proteins. That has to be the product of any of these systems. If any of them are correct, they're going to produce RNA. And they're going to have to, by definition, produce RNA that does not encode for a single protein. Not one. So in my search for the simplest organism, I found that viruses contain the least amount of proteins to become a viable, self-contained organism that, in the proper conditions, can reproduce. Is there a simpler organism than that? And it took me by surprise. I did not know that there are viruses that can exist without a protein coat. Not only that they do not have a protein coat and are able to survive and pass from one organism to another, their RNA, they're basically long segments of RNA, do not code for any proteins. They do not contain any information to make proteins to propagate themselves. These strips of RNA are called viroids. Virus void of protein information. They're called viroids. They're random nucleotides of RNA. But the crazy thing about them is they have the ability to enter into nucleuses, enter into membranes, and take over the replication machinery and make copies of itself and destroy the cell without a protective coat, without making proteins to replicate, to aid in its replication, or to encapsulate it to take it to another cell. This is the product that any origin of life system would have produced. And we find it today. I've always, and I mentioned in my book, if this quote-unquote self-replicating RNA molecule would have been able to replicate itself in the then non-friendly environment of at the origin of life, we should find them. If they were robust enough to be able to survive in those conditions, they should be represented when conditions became more habitable to life, because the earth eventually became changed by life to make it more habitable for life. We should still see those replicating, self-replicating molecules in the environment, anywhere. They would have been so mass-produced that they would have been able to survive in any niche that represented the conditions back then, which we still find today. There are plenty of environments where our carbon dioxide and acidic only, and we have organisms that live in those environments, those niches. But what we do find is these viroids that do exist in the quote-unquote conducive environment of cells, which again I claim, according to my theory, that is the same conditions we would have found in the oceans at the primordial soup in the Hadean era. So that to me should be the focus. At least that's the focus I take in my theory. These viroids, when you look at the sequence and map out the RNA nucleotides and you open them up, mimic the sequence, not the sequence, but the shape and the conformation of transfer RNA. Transfer RNA is the fundamental molecule that's necessary for life to work. Transfer RNA is the RNA that grabs the amino acid and allows ribozymes the ability to make proteins from an RNA template. Here's the other thing. The sequence of the RNA that is the heart of the ribozymes, the part of the ribozyme that cleaves the the amino acid away from the RNA, transfer RNA, is itself RNA. And when you open up and map out those nucleotide sequences and look at its structure, you will see that it is very similar to that of the viroids. If you have the viroid and you take away portions of it, you get transfer RNA. If you take the viroid's RNA and you multiply, you replicate it, and you get one of the most simplest errors in replication is the doubling of information or the doubling of the sequences. If you double it enough, you'll get ribosomes. So here is the key. What does RNA do? Which is one of the biggest discoveries in the 80s about RNA, is that they autocleave. They have catalytic ability. One of those catalytic abilities is they can cleave themselves. One RNA can activate and cleave a portion of itself to make it smaller, which would produce transfer RNA from a viroid. Replicate a viroid and get multiplication errors, and you make a larger and larger viroid, you get the harder ribozymes, which in many experiments have shown that if you remove all the protein coats, all the proteins associated with the ribosome, you still get the ribosome performing translation of RNA to proteins. So that to me is a clear winner. An informationless segment of RNA is the viroid. The viroid would have been the product of any origin of life system, whether it be emergent chemical evolution or inorganic material creating RNA molecules ad novum. You would produce a viroid. And the viroid, again, when you look at the genetic information of complex organisms, one of the first things that they produce is long strips of RNA that itself is cleaved by an RNA segment, an enzyme of RNA, which again is the same size as a viroid. This sequence of RNA's job in complex molecule cells is to take that long segment